Hi folks, in today's video we're going to be talking about cellular respiration, which as you know takes place in the organelles we call mitochondria. And it's important to remember that historically, evolutionarily, the mitochondria were originally free-living bacteria, heterotrophic bacteria. So it's best to think of the eukaryotic cytosol surrounding it as being cytosol, and it's also important to think about the interior of the mitochondrion also being cytosol, except that it has a different name, which we're going to call the matrix. Between those two spaces, between those two cytosolic spaces, you have a space between the membranes, and thankfully it's called the intermembrane space. So that's what we're starting with. Now, remember, we also have all of these folds in the membrane, and unfortunately, there's another word associated with that. So the vocab word is cristae for all the folds of the inner membrane, which provides us additional surface area to do the ATP synthesis. So, which brings us to the synthase. So the ATP synthase is a mega protein complex that generates the ATP from ADP and free phosphate. And as you might remember from the videos we've watched, this ATP synthase is driven, the turbine is spun, by the flow of protons, hydrogen ions, H+, choose your term. So these protons are going to be driving the spinning of the turbine by flowing down their concentration gradient, which means that somehow we're going to have to fill up the intermembrane space with a ton of protons. We need at least a tenfold higher concentration of protons in this space than in the matrix because we need to drive that spinning. So the process that's going to generate that proton gradient is the electron transport chain. So these are another set of protein complexes, and they are, again, going to effectively pump protons into the intermembrane space. And the shape changes that these proteins undergo, what drives their shape change to pump the protons, is by receiving and transferring electrons from member to member. So the first complex accepts an electron, goes through a shape change, pumps a proton, then transfers the electron to the next carrier. And each time it picks up an electron or drops off an electron, the shape changes, causing a proton to get pumped. That's the general idea here. Now, if we assume that the cytosol is pH 7, all right, remember pH is a measurement of proton concentration to some degree. It is the negative log of the proton concentration. So a higher pH indicates a lower proton concentration, and a lower pH is going to imply a higher proton concentration. So our highest proton concentration, hopefully, is going to be the intermembrane space. And so I'm going to label it as a lower pH, a pH of 6. This is not numerically accurate. Please understand that. But this is going to give you a relative idea of how we get those gradients. All right. So how do we get the proton gradient? Well, the electron transport chain is driven by electrons. We got to get those electrons from somewhere. The electrons, and let's just take a step way back, we're basically going to yank them away from food. Whatever fuel source we're using, whether we're talking about carbohydrates, fatty acids, or proteins, whichever source of food we're using, we're going to use something called the Krebs cycle to yank electrons away from them. And the technical term is to oxidize them. And we're going to take those electrons from, again, any fuel, carbs, fatty acids, or amino acids, transfer them to an electron carrier, which is just going to shuttle them to the electron transport chain. So let's not overthink what these carriers are. Yes, there are names for them. You need to know about them. Nicotinamide adenine dinucleotide, also known as NAD, is our key carrier here. There's also another one called FAD. But this is our just an electron shuttle. It's not doing anything other than being our sort of postal service for electrons. It takes the electrons from the fuel and carries them to the electron transport chain, diffuses to the electron transport chain. Okay, when we oxidize, when we take the electrons away from the fuels, they end up getting broken down. And you actually know the waste product of breaking down fuels. This ends up being the CO2 that we exhale. So there's the CO2 coming out. Again, as we oxidize an organic molecule, the waste product eventually ends up being CO2. Now, there are multiple fuels, as I've already mentioned. The one we're going to focus on is glucose. It's not the only fuel you can use. It just happens to be the most common in your body to be used as fuel for this process. So glucose is going to go through its oxidation process where electrons are removed. It's broken in half in a 10-step process called glycolysis to generate 2 half glucoses, pyruvates, which then go to the Krebs cycle. But again, don't forget the big picture. All we're doing is taking a big carbon-based molecule, glucose, fatty acids, or amino acids, oxidizing it, taking the electrons away from it, giving those electrons to a carrier, NADH, which carries them to the electron transport chain. That generates the proton gradient, which spins the synthase. Now, that's a whole lot of stuff to remember, but try to take a step back and get the order of operations here. All right. Overall, if we summarize all of that, this is aerobic cellular respiration, and it's going to generate a ton of ATP. This is a rather shockingly efficient process if you look at the actual sort of delta G's involved here. What we generate eventually is about more than 30 ATPs per glucose broken down. Now, that's going to be a whole lot more than an alternative that we'll talk about in a minute. Now, the one thing that we've completely left out of here is the whole aerobic part. Aerobic means using air. And when we talk about air, what we're really talking about is oxygen. So let's review the equation that you've memorized a number of times. Back in Living Earth, maybe even earlier, you memorized the equation for cellular respiration. The C6H12O6, the glucose, fructose, whatever it might be, being broken down to CO2, which is on this diagram right now. You can see it right there. But there's the other half of the equation, which we've conveniently left out, which is the oxygen. Now, this is a really important point, which is not easy to understand. The electrons passing from glucose by way of NADH to the electron transport chain have to go somewhere. The easiest way of explaining this is the electron transport chain is able to pump protons because it's going through shape changes. Every time it accepts and drops off the electrons to other carriers, it pumps protons. But the electrons have to go somewhere for the movement to continue. And our end electron acceptor, our true oxidizer in all of this is oxygen. All right, it's going to be the final electron acceptor here. If you add electrons to oxygen, making it effectively negative, and it binds some protons, you're going to end up with water. So that's why I've drawn in those little orange dots. Those represent the electrons. Oxygen turning into water is our final electron acceptor, and that's why the final equation hopefully makes sense. Now, I've certainly not balanced everything on this diagram purposefully. You don't want the balanced version other than what's at the bottom of the screen. But if you can understand that the electrons getting passed down the electron transport chain have to go somewhere, or else you get effectively a traffic jam of electrons, you've learned what you need to know for this class. Okay, so as I've mentioned before, the oxygen is going to be accepting the electrons, allows the electron transport chain to continue, allows for the pumping of protons, allows the protons to spin the synthase, and you get your ATP. So, what happens if there's no oxygen? This process is not the only process. What's drawn here is not the only way to make ATP. It is the most efficient way in our cells, for sure, and is the most common way in life, is what you're seeing on the screen right here. This is how we generally generate most of our ATP. But it is worth remembering that there are organisms out there that live in places where there is no available source of oxygen. And there are times in our bodies where certain cells may not have access to oxygen for short periods of time. For example, a sprinting muscle may not have access to oxygen or enough oxygen to do the whole process shown here. So there is a side process already shown here, and it's glycolysis. And glycolysis can actually occur in the absence of oxygen. Now, there is a trick to this. Glycolysis actually generates 2 ATP per glucose, which is not a lot. But the trick of this is the following. Just like the Krebs cycle, glycolysis is an oxidation process, which means it's going to yank electrons away from glucose, generating NADH. And as I mentioned before, we need to pass those electrons along to continue this process. In the absence of oxygen, all of the NAD plus is carrying electrons and is in the form of NADH. And in the absence of oxygen, if the electron transport chain is backed up with electrons, there is literally no NAD plus around. And that's a problem. If there's no NAD plus around, if all of it's in the form of NADH, there's no NAD plus to accept electrons. You can't oxidize glucose or anything else. So we need to have some way of regenerating the NAD plus. And I know this is complicated, but the big picture is that there is a side process, kind of a cheat code, to allow glycolysis to continue. And this is a way of effectively getting rid of the electrons and sending them back to another source, another acceptor. And so pyruvate actually can be used as an electron acceptor in a side process that generates one or another. There are lots of potential waste products here. You've heard of these. Lactate is also known as lactic acid. And this process happens in our sprinting muscles, our type 1 skeletal muscles. Another process breaks the pyruvate down into ethanol, also known as ethyl alcohol. And this is the brewing process or the winemaking process. These processes are known as fermentation, or more commonly, anaerobic cellular respiration. No air required. Most importantly, it can occur in the absence of oxygen. It does not generate a whole lot of ATP per glucose consumed, which means you're likely to run out of fuel really quickly, which is why you can't sprint for very long. You can sprint for a minute, but not much longer. But in the end, it does allow for survival in conditions where there's not a sufficient amount of oxygen. Okay, hopefully that gets you the big picture and also an insane amount of detail about the process. Feel free to rewatch, pause, and just watch little clips of this as needed, and bring your questions to class anytime you've got them. See you next time, folks.